So for over 16 years, I've had the absolute honor of working very closely with our next speaker, Bob Langer, and it is extremely humbling and my absolute pleasure to welcome Bob to the TLO's Golden Speaker Series this afternoon. Bob, for those who don't know him, is an institute professor here at MIT and a professor of both chemical engineering and biological engineering. Highlighting Bob's vast accomplishment really would in fact be a presentation in and of itself, so I'm going to name a few, and I promised him I would not embarrass him. So here we go. Bob in data. He's probably the most cited engineer in history, and as having authored more than 1,500 scientific articles. He's received over 40 honorary degrees from institutions worldwide. He's received over 220 major awards, including the Charles Stark Draper Prize, which is considered the equivalent of the Nobel Prize for engineers, the Millennium Prize, which is the world's most preeminent award for breakthrough innovations, and the Priestley Medal, which is the highest award of the American Chemical Society. Bob has been elected to the National Academy of Medicine, Engineering, and Sciences, and is one of 3 living individuals who have received both the United States National Medal of Science and the National Medal of Technology and Innovation. So we are in for a treat. Bob's like here at MIT works at the interface of materials science and biotechnology, and has a couple of core focus areas, including drug delivery based on lipids, polymers, and devices, as well as regenerative medicine, including tissue engineering and stem cell research. So in the theme of this event being hosted by the Technology Licensing Office, Bob is recognized actually as one of history's most prolific inventors. He is and the numbers actually really speak for themselves. I was looking at our database and was really thrilled to find that Bob's first disclosure to the TLO was actually in 1974. So Bob has been contributing to inventions for almost a half a century and has contributed to 720 distinct inventions that have been disclosed to the MIT licensing office. And those disclosures have given rise to hundreds of patents. And interestingly, as I looked at the data, his inventions actually stunningly reflect collaborations both across institutions as well as across disciplines, long before the concept of convergence was in vogue. His intellectual property has been licensed to dozens of companies, including about 40 companies that are considered startups out of MIT. And these companies address a variety of problems, real world problems, in fact. Some recent examples of companies include VitaKey, which works in the field of nutrition, Lindra Therapeutics, which develops resident devices for drug delivery to the gastrointestinal tract, Sigilon Therapeutics, which is developing cell-based replacement therapies for chronic disease, Frequency Therapeutics, which is tackling hearing loss, Tisium, which is using biomaterials for tissue reconstruction, and Swono Bio, which is using ultrasound as a delivery platform. So those are just a few examples of how broad the technology reach is from the Langer Lab. And without further ado, I'm happy to turn the stage over to Bob. We certainly look forward to hearing about his perspectives on his journey and how he approaches translation of technology and research to have impact in the real world. On a housekeeping note, we will have question and answer session at the end of the presentation, so think up some good questions, and if for those of us joining virtually, please feel free to put your questions in the chat. Bob, over to you.
Lauren, thank you so much. You know, I feel very privileged to work with the TLO, and in particular to have worked with Lauren for a number of years. You know, when people ask me about, what makes MIT unique and what makes Kendall Square unique, it's certainly because a large part of the TLO and because of Lauren and Lita Nelson and others that have worked there and have helped me so much. So I'm going to just actually start with a slide, the next slide. And, you know, sometimes people ask me, you know, how do I think about innovation and what advice do I try to give my own students? And what I try to do is try to dream up or do things that I hope will have a very big impact rather than incremental research. That's an easy thing to say, but you know, a lot of times when people go out, you know, to universities and they're so concerned about getting publications, you know, incremental research is a very good way to get publications. So it's not an uncommon thing to do. The second thing is conventional wisdom isn't always correct, you know, what you learn in textbooks and articles. The third is what Lauren mentioned, convergence, which is exposure and actually utilizing very different disciplines. The fourth is risk-taking. I think one of the things that's really made American industries what it's, you know, so amazingly good is that people do take risks and they do fail. Everybody fails. I've certainly failed a lot. I'll mention that. You know, in fact, Laura knows, like, I'm a big chocolate lover. One of my favorite stories is Milton Hershey, who, and I'm not even a Hershey chocolate fan. I'm not, but I love dark chocolate. But Milton Hershey tried to start a candy store about 8 times and he failed 8 times, but not the 9th, as I'm sure people know that it's still around. But fifth is, if your idea is important, expect lots of criticism. And finally, I would say never give up, you know, almost. I'm going to try to illustrate some of these points and talk about patents and other things that we've done with the TLO and about my own career. And on that note, you know, I graduated from MIT in 1974 with my doctorate in chemical engineering. And when I did, pretty much all my classmates went to work for oil companies. That's what chemical engineers did then. And I thought naively, I'm going to do what they do. So I got, you know, actually 20 job offers from oil companies, 4 from Exxon alone. And believe me, I wasn't that good. They just had so many openings. One of them really made an impression on me. I remember going to one of the Exxons in Louisiana, and they said, gee, if you could increase the yield of some of these chemicals by like these incredibly small percents, like 0.01%, they said that would be amazing. It'd be just so great and be worth billions of dollars. And I remember flying back to Boston thinking to myself that I didn't want to do that. And it really was a useful lesson to me after all these interviews, because it made me realize what I didn't want to do, even though lots of other people were doing it. And so one day I saw an ad to be assistant professor of developing chemistry curriculum at City College of New York. And I was very excited about that. And the reason I is when I was supposed to be doing my PhD thesis at MIT, I actually spent at least half my time helping start a school for disadvantaged high school students, poor high school students in the Cambridge area. You may not realize this, but Cambridge in 1974 had the highest dropout rate of any school, of any city its size in the country. And anyhow, so I got very involved, and it was also very liberal. You know, you didn't have to take math, you didn't have to take science. So I worked hard to try to make it interesting and relevant, and I thought I did a pretty good job. And so when I saw that ad to do really a lot of the same thing at City College, I thought that's great. I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea, so I found all the other ads I could to do something like that. I found about 40. I wrote them all. I don't think a single one wrote me back. I wasn't in the right box, I guess, you know, the chemistry education box. Even though I'd gone to MIT, which was a pretty good school, I didn't merit getting letters back from City College or other ones. So that wasn't so good. But it did make me think, you know, I wanted to use my chemical engineering education to help people. So I had another idea. Maybe health. I could work in that area. So I wrote to a lot of hospitals and medical schools. None of them wrote me back either. But then one day, I remember going into the lab where I was, Clark Holton's lab, and one of the older scientists said to me, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman. He said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But on the next slide, I want to show you what Dr. Folkman's idea was. He had this idea that if you could stop blood vessels from growing, that might be a whole new way to treat cancer. And this slide shows that. You know, the idea is that a tumor starts on the lower left and the cells become abnormal. It starts to grow as you follow those arrows in a three-dimensional mass. But when you get to the middle bottom, it doesn't get larger than 1 million cells. It runs into a nutrition problem. Cells in the middle of that ball can't get nutrients or get rid of waste. But he had a theory that the tumor solved that problem by sending out a chemical signal called TAF, or tumor angiogenesis factor, that caused the surrounding blood vessels, which you see on the bottom, to start proliferating wildly and connect up and hook up with the tumor. And when it does, that gives the tumor the nutrients and so forth. And then you see, as you follow the arrows on the upper right, the tumor gets bigger and bigger. And on the top, it's very vascularized. And it can spread through those blood vessels. That's a process called metastasis, which often kills. He had this idea that if you could achieve what he called anti-angiogenesis, which you see here, that maybe that would be a whole new way to treat cancer. Now, it turned out that most of the medical community thought this was a ridiculous idea, that diffusible factors didn't exist, that the tumors were more inflammatory and so forth. I didn't know anything about biology. Last time I've taken a biology course was in 10th grade. I thought it was a great idea. So he offered me a job. It didn't pay anything like the oil companies, but my job, he said, was to prove he was right, and to so doing, isolate the first blood vessel inhibitors. So one of the key aspects of that, to try to do something like that, was we had to have a bioassay to study blood vessel growth, and there really wasn't any. So we had to invent one. And our idea, if we could go to the next slide, was to use the eyes of a rabbit, which had big eyes, and you could put tumors in there. And then we needed a tiny delivery system that could deliver large molecules. All the molecules I was isolating were large molecules, and like, you know, proteins or sugars and so forth, polysaccharides. So at any rate, I needed this tiny little particle, like a micro or nanoparticle, that I could put next to the tumor and deliver these molecules for a reasonable amount of time. Now, that had never been done before, and in fact, there was hardly any work on drug delivery had actually ever been done by 1974. In fact, there was only one company in the world at that time, a company called Alza, that was doing any work on drug delivery systems. That was and they were all delivering small lipophilic molecules. Nonetheless, Dr. Folkman was on their board. So he went out there, he asked them, could they help us? And they had Nobel Prize winners in chemistry on their board, like Paul Flory and others. And what they told him, if we could go to the next slide, is that it's an impossibility. That if you try to put a large molecule in a tiny capsule, it's just too big to get out. It's kind of like asking any of you to walk through the wall, you know, right there next to you. So they just said, you know, forget it. But he didn't want to forget it, and I didn't want to forget it either, because we felt that without doing this, we would not be able to create that bioassay, and we wouldn't be able to, you know, solve the problem we wanted to solve. So I spent 2 years in the laboratory experimenting with different materials, different techniques, different theories. I actually found over 200 different ways to get this to not work. But eventually, if we could go to the next slide, I found one way to get it to work. And we were able to make these tiny little particles, like micro or nanoparticles. You see a picture of one on the left, cut in half on the right. And we were able to show, if we could go to the next slide, that you could use this approach to deliver molecules of virtually any size. And it was also the very first paper that showed you could release nucleic acids. For reference, you probably all know this, DNA and RNA are nucleic acids. We published this in Nature in 1976. I want to go back, though, about 3 to 4 months before that paper came out. So I was asked for the first time in my life to give a talk at a major scientific meeting in early 1976. I practiced this talk. It was a 20-minute talk for 2 weeks into a tape recorder. And I got up, and I was 27 years old, I remember, And I was giving this talk to this really distinguished audience of chemists and chemical engineers in Michigan. And I thought at the end of the talk, I actually did okay. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk, all these older scientists being nice people, they would want to encourage me, this young guy. But when I got done, I stepped off the podium and a whole bunch of them came up to me and they said, "We don't believe anything you just said." If you could go to the next slide, what they said was just what the ALSA people said. The molecules were too big to get out. They also pointed out to me that I was using organic solvents, like, which I was, ethylene, methylene chloride, ethanol, and they said that's going to denature any of these things like peptides, proteins, or nucleic acids. And actually, sadly for me, things went downhill from there. You know, I wrote research grants because I wanted to support the work, and my first 9 were rejected. And very soundly. I remember one of them came back from the NIH and they said, well, Dr. Langer is an engineer. He knows nothing about biology and even less about oncology. Now, I thought I knew enough about math to know that wasn't true, but I mean, not that I had any say in it. So I didn't get any of those grants. Also, I love being a postdoc. I really love being at Children's Hospital. I was the only engineer there. I learned so much. But you know, a lot of my friends, they start saying, Bob, probably you shouldn't spend your whole life doing that. And I did think, okay, well, maybe I'll apply for faculty positions. So since I'm a chemical engineer, I applied for chemical engineering faculty positions. And no university in the world would hire me. They actually said the opposite, or not the opposite of NIH. They said, well, an engineer doesn't do experimental bio work. I mean, you know, they should be doing energy or math, but the stuff that I was proposing made no sense to be done in a chemical engineering department. So I ended up getting a job in the nutrition department. I think I got it because the head of that department, Nevin Scrimshaw, a very famous MIT professor, he was kind of like what I'd call a benevolent dictator kind of professor. He, sometimes they have that at MIT. Nevin, you know, he was a great man, but he liked me, so he offered me a job as an assistant professor. But he didn't ask anybody else in the department what they thought, which might have been okay, except for one thing, which was the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice. Their advice is I should leave too. In fact, on the next slide, this is a couple of years ago, I got some award, and they asked Mike Marletta, he was a professor in the nutrition department along with me at the time. He ended up having a fantastic career. He was the head of Scripps. He was chair of Berkeley's chemistry department, member of the National Academy of Science. He was a very close friend, still is. And he made some comments about what it was like to be with me in the late 1970s. Here's what he said. He said, one evening I went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. I remember that well. But anyhow, that wasn't very good. So I had a very inauspicious beginning. I'm still here though, so it worked out okay. You know, you sort of put your head down and you keep trying, regardless of what other people said. So at any rate, I felt, you know, I could see with my own eyes, regardless of what other people said, that our drug delivery systems, you know, we'd put dyes in some of these little tiny particles and I could see them keep coming out. So, you know, we went ahead and started doing studies to see if we could find something that could stop blood vessels. That was the original goal. And by that time, I'd also isolated a whole bunch of different substances that I hope might stop the blood vessels. They were all large molecules. So working with a couple surgeons in Dr. Folkman's lab, Mike Klein and Ken Falterman, we actually tested maybe 100 different ones. 99 didn't work. We probably did 2,000 eyes. And the next slide's kind of gruesome, but let me just show it to you. This is the rabbit eye assay. We published this in Science. And what happens is if you don't put an inhibitor or have an inactive one, which almost everyone was, you get what you see on the left. In fact, if you didn't sacrifice these animals, which we do, what happens is the tumors become three-dimensional. They go out of the orbit of the eye, and they would kill. But we sacrificed them before then. But what was so striking is when we put this one inhibitor in, one fraction in, you get the vessels are lower, they're sparser, they avoid the delivery system in the tumor. And we repeated this 20 times, so it was quite striking. 60% of the time, these tumors never grew, and that never happened in the controls. So that was very exciting. It showed for the very first time that blood vessel inhibitors did exist, and would provide a pathway, like bioassays, that all future inhibitors could be studied. Now, it took medicine, and people may not realize this, but medical research is really slow from discovery to translation. It took 28 years from this paper before the first blood vessel inhibitor would get approved by FDA. But if we go to the next slide, what you see, and it may be hard to read, but one after starting 28 years later, 2004, one after the next blood vessel inhibitor gets approved by FDA. Avastin, that's one of the biggest, that was the first, and that is one of the 2 or 3 best-selling biotech drugs in history. And it's approved for many different cancers. Also, what you see on this slide is that these continue to get approved to this day, 3 this year. And they're all not only used for treating different kind of cancers, but they're also, actually, it turns out, the only pharmacologic treatment that exists for stopping diseases of blindness in the back of the eye, like macular degeneration or diabetic retinopathy. They've really revolutionized, the companies that have sold it. They've revolutionized treating diseases in the back of the eye and so forth. And so that was actually very exciting to see, but of course it took a long time. So going back to the 1970s, when I started doing this work at Children's Hospital and later MIT, Dr. Folkman, he was a visionary guy. He said to me one day, we should file a patent. Lauren knows I've got, quite a few patents now, but I had none then. And Children's Hospital had none then. And so we filed for what I understand to be the first time for a patent. And 5 years in a row, the patent examiner rejected it. So the head of the tech transfer office at Children's Hospital was a guy named Al Brazaghini. And he came down to see me one day and said, Bob, just stop. Keep trying to fight this patent thing. He said, you're never going to get it allowed. The guy's not that smart in Washington. And he said, just give up. But I don't like to give up. So I started to think, how could we convince the patent examiner to allow this patent legally, of course? And science really wasn't working. But as I told you in the beginning, when I first started doing this, everyone said it was impossible. It would never work. So I wondered, maybe somebody wrote that down. So next slide. So what I did in 1982, I went back. There's something you can do called a science citation search. So I looked at our 1976 Nature paper to see who referred to it. And I found a number of articles, but this in particular I felt was very useful. It's by 5 of the top material scientists in the world. And here's what they wrote. Generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, so surprising is a really good word for a patent examiner, I thought, that clearly demonstrate the opposite. So I showed that to Dr. Brazaghini. He flew down to Washington, showed it to the patent examiner, and the patent examiner said, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these 5 people that they really wrote it. So I was a young professor then. I wrote them, and they were all nice enough to write me back that they really wrote it. And next slide, and we got this incredibly broad patent. With that patent, I really wanted to see, and it means a lot to me to not just do the basic research and publish it, but to see it make a difference in human lives. So it turned out that after 10 years of working on this, I'd get a call from a multi-billion-dollar animal health company, and they wanted to use this and have me consult. And then the next year, I got another call from Eli Lilly. They wanted to use this and have me consult, and they were going to give me big research grants and consulting fees for the first time. And I was so excited about this, not just because of the money, which is very important to a young professor, to have research grants, but because I thought those companies were going to now bring what I did to help the world. But anyhow, they worked on these things for maybe 6 months to a year, and they didn't get exactly the results they liked, so they gave up. which was very sad to me. About a year or two later, Alex Klebanoff, one of my colleagues at MIT, said to me, Bob, he said, we should start our own company. So we did. Next slide. We started this little company, Enzotech. The first 4 employees were my former students. And we had actually, we're doing, I think, good science, but we didn't have a great CEO. And we were on the 5th floor at Sydney Street. On the 4th floor was a company called Alchemies, and they were trying to get drugs across the blood-brain barrier. That it was started actually by Alex Rich and Paul Schimmel, who were MIT professors, but they were trying to get it across the blood-brain barrier. But they had hired a very good CEO, Rich Pops, and but their technology wasn't working. So we did this merger. Today, that's a public company. It's got 25 products that are FDA approved or in clinical trials. It's got led to new treatments for schizophrenia, alcoholism, opioid addiction, and type 2 diabetes. They've got about 2,000 employees. Just to show you some pictures. So what happens is that Bidurion is every 2 weeks. So some of these were done by Alchemy, some with other companies that we taught, some other licenses that we did. So Bidurion is a treatment with a peptide for type 2 diabetes. Lupron Depot was originally once a month delivery system for a peptide for prostate cancer, advanced prostate cancer. Now Takeda, which makes this, is about every 6 months, and many, many others here. I'm going to talk about Gliadel in a minute, and also drug-eluting stents, which both came from patents at MIT. But anyhow, there are lots of these products, and they affect hundreds of millions of people every year. So let me now go to the second story I want to tell you. Next slide. So since I'd worked at the hospital, and now I'm at MIT, but I'd worked at the hospital. I still, by the way, have an appointment at the hospital, and I'm on the board of trustees at Children's Hospital. But, I became curious. How do materials find their way into medicine? And I thought naively that it must be older chemists or chemical engineers. And yet, when I looked into this, that was never true. Almost always, it was a medical doctor trying to solve a medical problem, and they'd go to their house and find some object that kind of resembled a tissue or organ they wanted to fix. Like the artificial heart was a lady's girdle, because it has a good flex life. That, by the way, was developed in 1967, still what's used today. And one of the problems is that the artificial heart, the lady's girdle materials, when blood hits it, it causes a clot, and the clot goes to the patient's brain, they get a stroke and they die. But to me, I thought, well, if something was designed to be a lady's girdle, it may not be the optimal blood contacting material. So my idea was, rather than go to your house, which is pretty much what everybody did in much of the 20th century, why not use chemical engineering design? Ask the question what you really want from an engineering standpoint, chemistry standpoint, and biology standpoint, and then design it from scratch. So we picked an example. Next slide. The example was that when we started, every biomaterial displayed bulk erosion. Bulk erosion looks like this. Over time, the material gets spongy and falls apart, which is okay for some drugs, but if you had insulin or an anti-cancer drug, it could just dump out, it would be fatal. So we said, from an engineering standpoint, what we really want is, on the next slide, surface erosion. And what we did, and I'm not going to go through the science because of the nature of the talk, but we asked different design questions. The next slide just shows what a polymer looks like. That's what all these were. And we asked different questions of what should be the molecular design? What should the monomers, the building blocks be? What should the bonds be? Even to the point of figuring out what ones, and we wrote these on the blackboard, even trying to figure out what would, at least theoretically, not be toxic and so forth, and metabolize and everything. We came up with this idea of a class of polymers that might be good, called polyanhydride, shown on the next slide. And our idea was that we would actually synthesize these polymers and because I was one of my good friends, Henry Brem, who was starting as chief of neurosurgery at Johns Hopkins. I'm sorry, starting as a young neurosurgeon at Johns Hopkins. He was interested in treating cancer patients. So we started not only to synthesize them, but to see if we could apply this to the brain cancer problem. But when we did this and we tried to raise money for it, it was almost impossible. Next slide. So when we first started doing this and we wrote grants, they were rejected time after time. First, people said, the reviews came back that you couldn't synthesize the polymers. But one of the great things is I had wonderful graduate students and postdocs. Here I had Howie Rosen, who was one of my students. He later became president of ALZA, the company I mentioned before, and he became president of it, and that became a $12 billion company. He later was elected to National Academy of Engineering. He synthesized them. So anyhow, they grants got still rejected. So the reviewers came back and then said, well, okay, even if you could synthesize it, these anhydrides are really reactive. They'll react with the drug. But Cam Leung, another one of my postdocs who now has a chaired professorship at Columbia and was elected to both National Academy of Medicine and National Academy of Engineering, showed there was no reaction. Then the reviewer said, well, okay, but they're going to be fragile, they'll break. But I had another couple of postdocs, Avi Dohm, who is now dean of pharmacy at Hebrew University, and Edith Mathiowicz, who's a full professor at Brown University and a member of the National Academy of Inventors, showed that you could make really strong polymers and they wouldn't break. Then the reviewers came back and they said, well, okay, but new materials are certainly going to be toxic. I had another graduate student, Cato Lorenzen. Cato's had an amazing career. He's a minority. He later became dean of medicine at University of Connecticut. He's been honored by being elected to all 4 national academies, and President Obama gave him the National Medal of Technology a few years ago. Then we sent the grant back after we showed it wasn't toxic, and they still rejected it. They said, well, the drug won't diffuse far enough. But I had another graduate student, Mark Saltzman, who at Yale University would later call up to ask to start their inaugural biomedical engineering department. He was the chair. And he's also now been elected to the National Academy of Medicine and Engineering, showed that it would diffuse far enough. Anyhow, this kept going on till 1996 when the FDA approved it. You can probably tell from the way I'm speaking that I'm very proud of all our graduate students and postdocs who've gotten incredible careers. The reviewers at the other universities, they haven't done that well. Just to show you a little bit of the clinical data, next slide. And this is from Henry Brem's group, that Gliadel, which is the wafer we use, it gives you, with another drug, really does prolong life. There is no treatment for this. No cure for this. And so we actually got it approved in 1996. It's been approved in over 30 countries, and it's still used today as one of the there's no good treatment for brain cancer, but it's still one of the major treatments for brain cancer today. Now, you might ask, after all those rejections, how did we ever get funding to do this? Next slide. So Howie Rosen, who I mentioned, he did his thesis on making these polyanhydrides, and we actually got a patent. And in 1985, this company Nova, started by Sal Snyder, a very famous neuroscientist at Johns Hopkins, the people came up to see me and they said, well, we'd like to license something from you. And I said, well, like what? And they said, well, anything, we'd like to, we have some funding, we'd like to see what would happen. So I said, well, okay, why don't you fund me to do the basic chemistry and then fund Henry Brem, my friend who was at Hopkins, to do the neurosurgery work. And it was great. Gene Whittemire was in the licensing office, and we did this license, and it was great, and we got all this funding. The only problem was, and I was so naive again, is Howie published his thesis before we filed the patent. Well, what's wrong with that? Well, what's wrong with that is that will make a U.S. patent not very good. So at any rate, a couple of things happened. So when Howie first started with me, we always wondered, did anybody ever work on polyanhydrides before? So now I'm going to go back before this patent was even issued. So I kept telling Howie, you know, you need to look at the literature, you know, and Howie started in 1981. So he looked back to 1970 and he found no articles on polyanhydrides. So I said, well, that's good, but can you go back further? Goes back to 1960. Still no articles on polyanhydrides. So I was thinking, this should be very easy to get a patent on. But then what he did is I said, well, go back a little further. And it's amazing. Starting in 1959, there all of a sudden are tons of articles on polyanhydrides. They stopped dead at 1959. 1959, you know, so there's no articles after 1959. But in 1959, 1958, in the 50s, there were a lot. They all came from one place. Next slide. They came from Japan, a Toyo Rayon company by a guy named Yoda. And so Howie said to me, you know, and we really always want to know what people did because it would really help us on our work. So Howie said to me, Bob, he said, I, you know, did all this work on literature review, and it turned out in 1981 I was going to Japan to give a lecture. I'd never been there before. He said, well, maybe you can find Yoda. And I said, well, that's a pretty big country. But anyhow, I was going to this conference, but it's a pretty big conference, and I, you know, you get these name tags, so I keep on looking at everybody's name tags. I actually found 3 people named Yoda, but they weren't the right guy. Unfortunately, or the very last day, you know, by this time I got very good at looking at name tags. And the guy sitting next to me at lunch, he wasn't Yoda, but he was from Toyo Rayon Company. And I said to him, that's the place that published this, and I said to him, do you know Yoda? He said, Yoda. He said, of course, he lives in New York City. So at any rate, I got back, I told Howie that, you know, and we called Yoda up, and he actually came to visit us at MIT. And I said to I said to him, I said, you know, he told us all his chemistry was great work and everything. And I said to him, well, why did you give up? He said, well, our whole goal was to make better clothes, you know, and we solved all these problems. He said, but there was one problem we could never solve. I said, well, what was that? He said, well, somebody would wear our clothes and they'd go out in the rain and they'd dissolve. I said, gee, that's too bad. But at any rate, we knew then that this would work, you know, and how we was, because that these were going to be degradable. So at any rate, so now, you know, as I mentioned on the last slide, nobody had ever used them for drug delivery. So we got this patent on drug delivery, which was going to be good in Europe because of their laws, but no good in the United States. And the people at Nova, the company, they were really angry at me. They said, well, you sold us this patent, and TLO sold us this patent, and it's no good. But I had another idea. You know, nobody also ever made them high molecular weights, you know, so they weren't going to be useful for many things. So what we did as Avidome, if we could go to the next slide, had come after Howie. I mentioned him as well. And nobody had ever made them over 7,000 molecular weight. So, and they weren't practical if they weren't. That was one of the points I made in that earlier slide. So they weren't practical if that would be the case. So at any rate, he made high molecular weight ones. We got this incredibly broad patent, like a composition of matter patent, and then the people were happy. And like I say, ultimately it got approved by FDA. One other story, which I think is really important on this, is, you know, we really made, with Nova, the company, pretty good advances. We got into phase 2, phase 3 trials, and it looked pretty good. But then this company did a merger, and they became bought by another company, by Sios. And Sios didn't care about brain cancer anymore, and they just stalled the project. And so Henry Graham, who was my colleague, and I, we talked to Lita Nelson, who was then head of the TLO, and we said, this is awful, this could help people, and they're just not doing anything. So, and our goal was never to care about the money, it was just, you know, to get used. So what Lita did, which was incredible, she talked to the company Sios, and she said, look, you know, we can, like sort of a carrot or stick. If you develop it, she said, that's great, we won't do anything. But if you don't, we're going to try to take it away, but you know, from you, and we, you know, but what we're willing to do is, if you guys come up with a way to develop it, start a new company, we'll really lower our license fees a lot, maybe by a factor of 10. So what they did is that Nova, they started a spinoff called Guilford to develop this, and they, took it through clinical trials, and like I say, it got approved in 1996. It's still used 27 years later. Let me go to the next story I wanted to tell you. This was about inhalation, and this was work from David Edwards, who was a postdoc of mine, and so before David, everybody always designed what I'll call porous air, large, tiny aerosols. The thing is that everybody always thought aerosols had to be like 2 microns in diameter or less, and the reason they thought that is that if they were bigger, they'd settle in the back of your throat. So, and the problem with that is that when you make them small, you're lucky if you get 2% from the device into the patient. And so it's kind of like wet sand. They're so small, they stick together in the device in the air. By the time you get to the lung, you don't hardly have any. So everybody always, other than us, tried to solve it by changing the inhaler, to have more force to, get like mechanical engineering to break apart the sand-like aggregates. And that worked somewhat. You could go from maybe 2% into the lung to 3%, which still is going to make the device 50% smaller and the cost 50% less. But David Edwards, when he came to work with me, had a radically different idea. I suggested to him one day that lung delivery would be a really good thing to work on. And so we thought, well, nobody ever changed the aerosol. So we did. And our idea was to make it super porous, but much, much bigger. And he showed by modeling that aerodynamically, if you did that, it wouldn't settle in the back of the throat, even though it was bigger. But because the surface area is so much less, per unit volume, they shouldn't stick together, right? Like wet basketball, or not much. Wet basketballs won't stick together like wet sand. Also, normally when you give an aerosol, they don't last long because macrophages eat it. It's like eating a small meal. Now, that's phagocytosis. Now the idea was that it should take a lot longer because it's like looking at a big meal. So we made some of these, some of our students, next slide, they made aerosols. Just to give you a sports analogy, before us, everything looked like little golf balls or little baseballs. Now, ours were like big wiffle balls. And this is a SEM. And then if you do what's called an Anderson impactor, this is an in vitro test, next slide, you get 10 times more, not just a tiny percentage more, 10 times more into different lobes of the lung. And next slide, these are human experiments that David did. They can actually last for, for several days. So that was very exciting. We published this in Science. And we also, again, got a patent, next slide, which the TLO did, and is very, very broad. And next slide, we started another company with quite a few of our students, and it did very, very well. David was president, and Terry McGuire, a venture capitalist, actually did a lot of the business work. And we got into the clinic right away, and the investors made over 1,000% IRR. And David even was able to start a nonprofit from it. And today, next slide, just to show you one picture, this is, I think the principle is being widely used, but this is a treatment for Parkinson's. This is a tiny little simple inhaler that you just press. Before this, you could never do more than 2 mgs for something that size. This does 70 milligrams, and so it's been quite remarkable. Let me go to the next story I wanted to tell you. So, a lot of people started companies and proteins, proteomics, on nucleic acids, genomics, but nobody really ever started a company on polysaccharides. And I had a couple of postdocs, Ram Sesasakaran, who's now a full chaired professor at MIT, and Ganesh Venkatraman, and they worked with me and Charlie Cooney, and we thought, well, let's start a company with Ram and Ganesh on polysaccharides, which are much more complex because they're mixtures. So our idea, next slide, was to harness what we call glycomics to create new therapies or existing therapies for human diseases. Now, if you, just to give you an idea of of what a polysaccharide is. Heparin, which you may know of as, that's a polysaccharide. It's actually a mixture. You know, if you have insulin all is the same molecule. Heparin is not. It's different molecular weights, different substitutions, and so forth. But it's approved by the FDA. So one of our thoughts was, next slide, if the mixture's approved, why couldn't we take individual molecules out by fractionating them and then sequence, and then could we actually sequence them for the first time? Nobody had ever sequenced them before. But Ram and Ganesh actually came up with a way to do that, which was published in Science. And without going into the science too much, it involved it involved using a molecular scissors to cut these. That's enzymes like heparinase, which I published in Science, coupled with using mass spectrometry to identify the sequences. And then Ram and Ganesh developed an algorithm to put them back together so that you'd get the whole polysaccharide, and patented that. And next slide, we thought we could make new heparins, other complex polymers that we could sequence, and even new glycoproteins. So in 2001, we started Momenta with Ram and Ganesh. We got funding from Polaris, we went public, and we got many, many deals. In 2010, you know, it's interesting when you follow these, if you look at stock analysts, you know, it's very interesting. They kept saying, well, Momenta is going to do terrible. They're competing against all these big companies like Teva and others, and nobody, and everybody's trying to make Levinox, which is a low molecular weight heparin, biogeneric. But the problem was, you know, even though, you know, we were this tiny company, we had the patents and we also had the know-how. So even though all the analysts said Momenta was going to go under and do terrible, it didn't. We were actually the very first company to get FDA approval. It was actually the first complex drug ever approved based on analytic data in history. And it was the largest syringe launch in history. We also would get a Paxone approved and had other programs. And in 2020, Johnson & Johnson acquired Momenta for $6.5 billion. Just to show you a few pictures. Next slide. This is enoxaparin, which is the low molecular weight heparin. When that got approved, next slide, Momenta did pretty well. But I want to tell you one other story. Next slide. In 2008, 15 years ago, a lot of people started dying all over the world from contaminated heparin that came from China. These are just a few headlines, and they kept going on and on. And I had once been head of the FDA Science Board, their highest advisory board, for a number of years. You know, it's not a full-time job. I was still at MIT. Ram, who I mentioned, he had also helped the FDA. So they called us up. They said, could we help? And so what we did is pretty unprecedented. We did this work at MIT, along with Bob Linhart, one of my other former postdocs who was at RPI, with Momenta, which had the sequencing technology, and with the FDA and the CDC. And what we learned, next slide, would stop that contamination. The bottom paper was published in Nature. We found that the contaminant that was killing these people was oversulfated chondroitin sulfate. We also published 2 other articles in the New England Journal, which is the most widely cited journal in medicine in the world. And after this, there were no more deaths. So one other story I want to tell you is that we could, I mentioned early on that we could make microparticles and nanoparticles. When you make these particles really small, they work, but a lot of times they stick together because, again, they're so tiny. Also, macrophages can eat them. So one of the things we also did is we took polyethylene glycol, which is going to attract a lot of water. It's actually FDA approved itself, and used it to coat little nanoparticles. And that really was revolutionary. It would change how long these would last, and it would prevent them from sticking together. And then Omid Farrokhzad came to our lab, next slide, and he even put targeting molecules on these. I think a lot of the time I don't explain this very well, so I thought I would show you a video that Nova, the TV show, did when they came to talk to me about our work.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should tell you that a lot of the clinicians I work with tell me it doesn't really blow up the cell like that. But at any rate, you get the idea. And what was really exciting is you can not only use this for small molecules, but as I'll get to, siRNA or messenger RNA. And a number of companies that I got involved in helping get started use this, and we kept Lauren very busy over the years. Next slide. So the first of these was Accusphere, and Accusphere was going to use this for creating new imaging agents. They were a public company. Bind was going to use it for, for new cancer treatments that they've since been acquired by Pfizer. Selecta was going to use it for, different ways of basically getting immune responses to different enzyme treatments. They're a public company and are in phase 3 trials. Alnylam actually was started by Phil Sharp and others, and I was on the founding scientific advisory board, and Akinna Kink, who was one of my students, was one of their first employees, and he led a whole effort, if we could go to the next slide, to create Onpattro, which is siRNA in a lipid nanoparticle with a targeting molecule, just like I showed you in that video. And one of the stories I wanted to tell you on this is that, we worked a lot with, I was on their scientific advisory board. Dan Anderson was a postdoc with me. He's now a full professor. But Dan had done a lot of work in our lab on creating new lipids, and Alnylam wanted to license that. And of course, we wanted to get a grant. And I still remember that Lauren and Lita Nelson, they spent so much time, I'll be eternally grateful to her and Lita for that, negotiating with Alnylam. And of course, part of the thing, and a lot of schools don't realize this, a lot of schools, what they're concerned about is getting money for the TLO and so forth, and universities judge things that way. But we wanted to get a grant, because we thought we could advance the work. And so I still remember, I think I got a call from Lauren at 1:30 in the morning one night. She said, well, Lita and we decided we'd sit with the Alnylam CEO and CTO, or I'm sorry, COO, until we reached a deal. And we finally got the deal, and you guys are going to get, something like $10 million for research money. And that was great. We published all these papers in Nature, and I think it advanced the field tremendously. And Dan has done unbelievable work. And, and he's not talking today, but Dan will later start CRISPR therapeutics and other ones. And again, using principles that would come out of here. And this actually was, for Onpattro, was the first, siRNA drug ever approved. And finally, if I could go back to the last slide, just go back one. So Moderna. So so by 2010, I could certainly see that some of these various things we were doing, delivery systems could be very useful. And Derek Rossi, who was a young scientist at Harvard, came to see me, and he, myself, Ken Chen, and Nubar Fayan, who started Flagship Pioneering, also is an MIT grad, actually, in a corporation. Now, why don't we go 2 slides forward? We had this idea to start a company to make proteins, but to make them out of messenger RNA. And that had never been done before. And of course, again, you're going to need nanoparticles to protect them, and pegylated nanoparticles to boot. We also thought that not only could you, and the beauty of this is that normally it takes maybe a year to make a protein, or you use eggs. This you could make almost immediately, because if you had the messenger RNA, you just give it to the patient, and the patient will do all the work. She'll make the protein. Also, you could treat diseases you could never treat before. Next slide. Like ones that were intracellular or membrane bound. Now, of course, the story I'm going to tell you, some of you may know, or at least parts of it. So Moderna, we started in 2010. For years, the press, stock analysts, and everybody ridiculed Moderna. They call it like Theranos, and there's this paper stat, and they would ridicule it, say this would never work, and so forth. But still, by the end of 2019, we had 13 products, including 8 vaccines in clinical trials, and then COVID hit. Next slide. So this to me is, I think, quite exciting that here the Chinese scientists published the sequence of the spike protein in January 11th. It took less than 2 days for Moderna scientists to make the messenger RNA and design it. Put it in nanoparticles and ship it to our clinical partner NIH for testing. And the first patients were treated, just another month or so later. So 2 months from really start to doing this and doing what we call a phase 1 trial, which is to look at the highest safe dose. Within 2 months, we had that data, and of course, you're a public company, you have to announce it. And the data, I thought, even though it was on a limited number of patients, was really good. It showed that you got antibodies and so forth. Nobody, people were safe and everything else. So I would have liked to have thought that the world would be excited about this. The news media would be happy about it. Scientists would be happy about it. But let me go to the next slide, which was published 2 months later. This is May 19th of 2020, and we have our local paper, and their front page headlines is this is not how you do science, and you can see those pictures on the front page, and so it's pretty discouraging. Nonetheless, I want to go 6 months after that. 6 months after that, we had done what's called the phase 3 trial. That's 30,000 patients, and if you look at the 30,000 patients, 15,000 are treated, 15,000 placebo. And I'm on the board of Moderna, still am, and we broke the code. And it was pretty stunning. Not a single person died, not a single person went to the hospital, and it was 94% effective. And the next slide just shows a picture of it. And just to show you some of the clinical data, next slide, you see this, and these are all within the past year or so. The enormous difference in hospitalizations between unvaccinated, vaccinated with no booster, vaccinated with a booster. If you look at the efficacy, next slide. The Moderna is the best one, 94%. Pfizer and BioNTech, also using pegylated nanoparticles, 91%. Johnson & Johnson, which is a different kind of vaccine, 66%. Let me show you a little bit more from published less than a year ago in the New England Journal of Medicine. Next slide. And you see with the 2 pegylated nanoparticle mRNA vaccines, you see really very, very low incidence of infection, the lowest being, again, with the Moderna mRNA 1273, even with close to 400,000 patients. And the next slide is over 400,000 patients. And notice that even after almost half a year, you get about 0.06% hospitalizations. Very low hospitalizations, very low deaths with this vaccine and with the BioNTech one as well. And of course, now we know that it can be useful for other things. Moderna announced about a month ago, next slide, that you can actually create what are called personalized cancer vaccines. This is really quite remarkable that you have now patients with advanced melanoma, but you can look at the patient's cancer. You get a biopsy and you can find mutations, and you can make it just like you attack the spike protein. Now you can attack those mutations, make mRNAs that will attack the top 34 mutations, put them in nanoparticles. And this trial was to take the best known drug, immunotherapy drug, Keytruda, by itself, which is the standard of care, and then with the personalized cancer vaccine. And what you can see is the risk of recurrence or death is reduced by 44%. And again, Moderna announced just a few days ago, again, very substantial success against RSV. So the last thing I thought I'd talk about is tissue engineering, which is another thing I've kept Lauren busy on. Just briefly, next slide is a little baby why don't I go to the next slide? But that baby was treated by Jay Vacanti, who is my collaborator. He's at Mass General now, was at Children's before. And we had this idea that you could take almost any cell type. Also, you could take stem cells, but people didn't talk about them when we first proposed this. And you could grow them in a bioreactor on a polymer scaffold and virtually make any new tissue. The next slide just shows a picture of some of the scaffolds with cells on. And the next slide just shows that really you can use ultimately CAD-CAM techniques to make a nose in this case, but you could do anything. And and design these in any way. So one of the things that got approved, next slide, is new skin. Here's a 2-year-old baby badly burned, but you can take the product, next slide, put it in the child at the time of injury, next slide. But let's come back 3 weeks later, next slide. And now a month later, I'm sorry, 6 months later, next slide. And you can see he's healed. And this is now approved by the FDA for patients with skin ulcers and burns. And it took 10 years for us to get a patent on this. The next slide just shows that. And again, Lauren has, this was licensed to different groups. You can also use these approaches not only to make new tissues and organs, but to create tissues and organs on a chip. And the next slide shows what some of our students, Gordana Vanyak and Milika Ritzik did. And you can take heart muscle cells and you'll get sarcomeres and intercalated discs. And I'll show the next slide, just a video of the heart on a chip. And you can see it just beating and beating like this. 6 million cells. Lisa Freed was also very involved in this. And it keeps beating and beating and beating. And you can actually use this for like an EKG on the next slide, or for drug testing, which companies are now doing. And on the final, or next to final slide, I was just going to show you a list of some of the ones we've been doing. And again, keeping Lauren busy. Well, skin is on the right. Heart is it was what I just went over. Alice Stanton is working on creating a brain on a chip with Li Wei Tsai, who's one of our collaborators. We've been working with Steve Zeitels. Now I'm going to the top. You see a picture of me with Julie Andrews to make new vocal cords. Pancreas, that's Sigalon, which Lauren licensed those little capsules to them. Blood vessels, Laura Nicholson, who is one of my postdocs, started a company which is now public called Humacyte. They're in phase 3 trials. You could do liver, cartilage, spinal cord's been licensed to in vivo. Hearing, she mentioned, has been licensed to help start frequency therapeutics. GI tract on a chip, been licensed to Vivitex. Gio Traverso led that work. So there's all kinds of things that are going on, and there'll be many more in this area. And finally, I just wanted to say, when do we start a company? Sometimes people ask that. And on the final slide, though I'm going to make a few other comments, what is almost every time we've done it, it's a platform technology. Like you could use this for drug X, drug Y, drug Z. Pretty much all these companies have done that. They make products. We've published almost all of these in top journals like Science or Nature. And as you could see, we've really made a premium of getting great patents, seminal patents. And the TLO, again, has been great about hooking us up with really great people, really good lawyers that can do this, and then licensing them out in ways that really maximize their use. We also like to have a in vivo proof of principle, and pretty much all these, there are about 40 companies we've been involved in starting, they've been done with my students. And our students will walk through walls to make these things work. A number of them have become CEOs and CSOs and so forth. Lauren asked me also if I might just comment a bit on both working with the TLO and also sources of funding. Well, I think we keep Lauren busy all the time, and all the people who help her and work with her, and they've been just terrific. I mean, I always say that MIT is the best technology licensing office in the world, and that really is, I think, key to getting these companies launched. I've actually been asked by 4 different governors to help them and give talks. I think, Virginia, New York, Illinois, and others. And this, everybody wants to have Kendall Square in their backyard. And really, I think this is having MIT and having a great TLO and it has been important. Also, Lauren asked me if I would mention industry research. I gave you the example of Alnylam. Gio Traverso and I have also had done a lot of work with Novo Nordisk on creating pills that you can swallow that someday will deliver insulin. That's now in clinical trials from work he and I and our students did. And they've licensed the work. And also, foundations have been terrific. We have had a 10-year relationship with the Bill Gates, who came to see me, you know, they've given us a great deal of funding, and they've also given companies we've started a great deal of funding. And so, and the Juvenile Diabetes Foundation was the one that gave us the funding that enabled Sigilon to start. So those have been great. Sometimes they may be a little tougher for the technology licensing office to work with, but somehow they still do it with a smile on their face. So thank you for having me. It's really a pleasure to speak. I hope Lauren won't be too bad at me for going over all this. So anyhow, thank you so much.